Dear audience, the topic of my PhD is the role of gut microbiota in cystic fibrosis. My name is Blanka Bódy. My vision is to offer a complication-free life for people with cystic fibrosis by investigating the role of gut microbiota in the disease. So I have two specific ongoing projects to achieve this goal. The first project is about the comparison of gut microbiota in cystic fibrosis patients and healthy individuals. So this is a sketch of the gut wall of an otherwise healthy individual and a CF patient. When the CFTR channel works just fine in an otherwise healthy individual, it is dysfunctional in a cystic fibrosis patient. It results in several systematic and local complications, which could all potentially induce dysbiosis. So our aim was to compare the gut microbiota of CF patients and otherwise healthy individuals. So we included articles with all age groups, comparing CF patients and non-CF individuals. The outcomes that we looked for were alpha diversity, beta diversity, and the abundance of certain bacterial taxa. At the end of the selection process, we ended up with 14 eligible articles, and after the search update, one more article was eligible. The outcome measures that we used were standardized mean differences with 95% confidence intervals. We could include 3 alpha diversity indexes and some relative abundance data to the meta-analysis part. All the other outcomes, 2 alpha diversity indexes, beta diversity, and absolute abundance went to the systematic review part. So let's jump to our results. First of all, about alpha diversity. To put it the simplest, it measures how many bacteria there are in a given sample and how evenly the different bacterial taxa are distributed in that given sample. And there are many indexes that help us measure alpha diversity, and Shannon index is one of these indexes. So as I've mentioned before, we calculated the standardized mean differences between groups. And we divided the population into 3 subgroups according to age: infants, children, and adults. If the articles lie on the left side of the line of no effect, it means that CF patients have a decreased gut microbiota diversity compared to controls. If they lie on the right side, it is increased. So let's jump to our first subgroup, infants. On the first look, we do not see any difference in the infant subgroup between CF patients and controls. However, when we look at the 2 other subgroups, children and adults, CF patients in these subgroups have a significantly less diverse gut microbiota compared to controls. The second alpha diversity index that we measured was observed autos. It only takes bacterial richness into consideration. It ranges from 0 to infinity, with the higher numbers indicating the higher diversity. The 2 subgroups here are children and adults. And looking at the diamond in the adult subgroup, we can see that CF patients had a decreased alpha diversity compared to controls. However, this difference was not significant. The third and last alpha diversity index that we analyzed was SHA1 index. It takes richness and evenness into consideration as well, but it puts more weight on evenness. 2 subgroups here were adults and children, and looking at the diamonds in the adult subgroup, it clearly tells us that CF patients in this group had a significantly less diverse gut microbiota compared to controls. So let me refer back 3 slides when I said that there was no difference in the infant subgroup between CF patients and controls. We wanted to take a closer look on this matter, so we calculated standardized mean differences for Shannon index in the infant subgroup between people with CF and controls during the first year of life at 4, 6, and 12 months of age. Seeing the position of the diamonds, we can see that there is a decreasing tendency of alpha diversity in people with CF compared to controls during the first year of life. We also displayed it on this figure. It might be a bit easier to understand. So the different colors represent different studies that we included. The straight line represents the CF group, and the striped line, the control group. So let's jump to our results about relative abundance. It is a percentage measure. It gives us the percentage that a given bacterial taxon takes up from the whole bacterial community in that given sample that we measure. So the two taxa that we could meta-analyze were Bacteroides and Veillonella, both in the infant subgroups. And looking at the position of the diamonds, we concluded that there was no significant alteration in the abundance of bacterial taxa in people with CF compared to controls. So the strength of our analysis is that we could analyze age-related differences and the gut microbiota maturation during the first year of life. And we have a higher evidence level due to overall larger sample size. However, our included articles had a high risk of bias, and we unfortunately had limited data on abundance. And the included articles only represent high-income countries. So in conclusion, CF serves as a barrier to the diversification and maturation of gut microbiota, with the implication of research that we should put way more emphasis on investigating the therapeutic microbiota modulation, as well as CF-related factors' influence on gut microbiota composition. So we have submitted our article 3 times, got rejected 3 times, so the 4th time we will submit it to Clinical Medicine, hopefully this week. My second project is the effect of CFTR modulator therapy on gut microbiota in CF patients. So CF comes with several gastrointestinal manifestations, which includes dysbiosis. We have two core problems that could potentially influence dysbiotic changes. The first one being the CFTR gene mutation itself. It results in insufficient CFTR protein level or function. However, we have a solution for this problem so far, the CFTR modulator therapy, which acts by either increasing the folding and trafficking of the CFTR protein to the cell surface, or enhancing the function of the CFTR channels, the inactive CFTR channels. The second core problem is a bit more complex one. It is all the CF-related factors' influence on gut microbiota that could potentially induce dysbiosis, namely exocrine pancreatic insufficiency, high-fat diet, constant medications, low motility, and so on. So our question was, to what extent is modulator therapy effective in resolving dysbiosis? We included only children with CF before and at certain time points after initiating CFTR modulator therapy. Our outcome measures were the same as for the previous study: alpha diversity, beta diversity, and the abundance of certain bacteria. And our hypothesis is that dysbiosis is reduced after the initiation of modulator therapy. So the source of data was the cystic fibrosis-related pancreatic disorder registry at National Pediatric Institute. We conducted database filtering, then data cleaning, then microbiota analysis took place at the Institute of Medical Microbiology, and now we are at the state of biostatistical analysis. We included 29 patients at 4 time points: baseline, 6, 12, and 24 months after the initiation. And I also brought you a preliminary figure, a very preliminary figure about the prevalence of bacterial phyla in our samples. So this figure might seem a bit overwhelming at first, so let me break it down to you. At the left side, you can see the follow-up points where we looked at the samples. At the bottom are the CF subjects themselves, one by one. And at the right side, you can see the bacterial phyla that we could detect in all these samples. The different colors only indicate different patients, and the bigger the bubbles, the more abundant that phylum was in that given sample. So in my 2 projects, I aim to investigate the role of gut microbiota in cystic fibrosis. I wrote the quote, "The man who moves a mountain begins by carrying away a small stones." Thank you.
So I would like to ask you, for your first project, why did you have to use standardized mean difference for your
Great question, thank you. Mean difference, your question, does it refer to why is it standardized or why is it mean difference?
Why is it standardized?
Oh, okay. So as you saw, all the studies use the same measure, so that can be the source of confusion. We standardized the calculation because these calculations can be influenced by the difference in the detected phyla. So if different bacteria were detected in the different articles, it could influence the measurement of these indexes, and also if the distribution is different, that could also influence the results. So that's why we thought together with the statisticians that it would be good to standardize these mean differences.
Yes, the reason I was surprised is because specifically for Shannon index, it takes into account the proportion of the bacteria already. So,
yeah.
But I understand. Thank you.
Thank you for the question.
Why microbiome and CF? It was not clear for me, so why these questions are come up from the literature, because microbiome is a big weapon and it's a big debate actually. So everybody is thinking, is researching, but it was not well discussed connection between CF and gut microbiome already. Can you tell me why you choose this topic?
Yeah, of course. So there's an extensive research going on about gut microbiota in CF, as you have already mentioned. Our question really was, so why this topic seemed important for us is because there are certain discussions about dysbiosis potentially inducing or worsening certain conditions, for example, malabsorption or some intestinal complaints, for example, diarrhea or abdominal pain, or it is also linked to certain diseases, for example, GI cancer or IBD, IBS.
Sorry to interrupt you, that this is a cause or a consequence of the disease. What do you think?
Yeah, well, this is a great question. It is, I won't be able to answer this question directly, so it can be both, of course. So it can also come from the CF-related factors, so the low intestinal pH, the low motility, the thick mucus, and so on and so on. But dysbiosis could also further induce these dysbiotic changes. So
the microbiome data from the literature is from the normal sequencing or from the shotgun new methods? What do you think, which is better? And do you mix these both results, or?
This is a great question. So we mix these results. So they are both from whole genome sequencing and both from shotgun sequencing, so 16S rRNA sequencing. And I know this is a confounding that we mix these results, but otherwise we would not have enough data to compare these two groups, the CF patients and non-CF individuals. So that's why we did this.
Your first paper is in the review status,
yeah, we will submit it.
You already submitted?
Yeah, this week, or I hope so.
So thank you very much. And then I'm not sure if it is anyway turned on, but does it make any difference?
For recording.
Oh, it's for recording, but if it is turned on, anyway. So my first question, so it's very nice, and then I like the concept. So with the meta-analysis, I have a few questions. What might help you for the initial step? The number one, that you say that in infants there is no difference in the composition of the microbiota, which surprises me, unless in all these literatures they excluded the formula-fed infants. So it is just breastfed infants in this study? So then how it comes that there is no difference?
As I've shown you on the, I don't know which.
So I know that it's a published data, somewhat surprising that there is a study where there is no any random difference.
Yeah, so most of the articles detected some kind of difference. It was not always a significant difference in all the articles, and all the articles that I can think of now that we included both formula-fed and breastfed infants in this subgroup. So, and we could not really account for this difference because they did not report which infants were formula-fed and which were not when they reported the results. So that's why we could not differentiate between these groups.
And in your opinion, the choice of the infant feeding would somehow
Of
affect
course.
the disease out there?
Of course.
Yes.
So this is also a confounding.
Yeah,
Yes.
exactly. And then the other idea that in these very small babies, how do you know that they really have the disease? So do they have already the clinical symptoms, or it is based on genetic testing?
So not all articles reported if they already have symptoms or not. I think all the articles that are included here did genetic testing to begin with.
Yeah,
so,
I assume so, because they are too small for that.
Yeah.
And for the second project, what is your patient population? What is the age of the patients?
It is a big range. So it is from 2 years of age till 18, 19 years of age. It is a big cohort, so we plan to account for the age differences, which is not that simple because we have only a small portion of the patients here in this analysis. So 29 patients is not that much, and if we also have to divide them by age, that is, well, a limitation for us. But we will do that analysis.
They all have to have the disease, that's right. Yes?
I'm sorry?
So they all have the disease.
Yes.
Besides cystic fibrosis.
Yeah.
Okay. Yeah, that's... I see. It's difficult. Yes, but maybe you do a follow-up. What do you think if you use this small patient number, but you make a follow-up at how the microbiota is changing, and then it might give you a second good research insight, which will overcome the limitation that you don't have so huge number of patients. Yeah,
that would be awesome. The only thing is that we have many patients in our cohort so far, so 135 patients, and only 29 were eligible for this analysis because they were not the most enthusiastic to give fecal samples upon visits, which is of course understandable, but I'm not sure if we continue on with the follow-up, we will have that much more samples.
Okay, thank you and good luck.
Thank you.
Yeah, thank you very much. A very interesting talk and interesting topic.
Thank you.
I would now, based on the comments of you, rather refer to other parts of the disease. As you've mentioned, it's kind of influencing the permeability of the membrane, and obviously the gut is one organ where you see effects, but the main clinical problem leading up to lung transplantation is in the lung. So I was wondering whether you have included any information on comorbidity, but also on co-medication, because there's one type of medication that is frequently given in cystic fibrosis, which I think could widely influence the microbiome in the gut.
So this question refers to my second project, right?
To the first one, because I do not know whether there's maybe information in the literature, and then in the second one, whether you on your own track it. Yeah.
Okay, so let me start with the second one. That is easier. So we have put emphasis on not to include patients with comorbidities. So if they have CF-induced liver disease, or if they had diabetes, which was induced by CF also, we excluded these patients. Of course, we could not account for the state of the lung involvement in every case because they all had some type of lung disease at this point. So that is also a factor that we will have to take into consideration. As for the first project, most of the articles included patients if they were not on constant antibiotics 2 months or 1 month leading up to the trial, to the analysis. And we also tried to include only patients, only studies who included patients with no other comorbidities that could influence gut microbiota. So there was, for example, an article with patients who also had reflux disease, so we excluded that article. So we tried to take this measure into account as well.
So as homogeneous as possible, because obviously they receive antibiotics, yes, those patients, some a lot. So do you think this could influence this age difference that you see, so that the very young children have not received so much antibiotics, which
yes,
is why...
Of course. So the newest articles about the differences in gut microbiota in the CF group all come to the conclusion that the factor that mostly influences gut microbiota is not the disease itself, but antibiotics given to these patients. So, of course, it does influence microbiota as they age. That is also a reason why there's a difference between these age groups.
And are there differences in the antibiotic regimens that maybe have been used? So I'm not an expert, so maybe there are national guidelines that differ?
Well, these studies were all from high-income countries, which I think all refer to the guideline at the when they treated their CF patients. So the guidelines strictly say what type of antibiotic regimen should be given in which case. So they mostly use the same antibiotic regimens. Of course, if there was an exacerbation prior to the study, it was another different antibiotic regimen from the patients that did not have an exacerbation, were only on azithromycin for the long run. So that could also differ, yes.
So now my last question goes to the impact. So do you think, based on this information, your work could influence, for example, the antibiotic regimens to be used because you see differences, or any co-treatment like probiotics or something that would positively influence
well,
disease?
We have known so far that antibiotics do influence microbiota composition, so I don't think that this study would be such a big breakthrough on this field, but I do think that seeing these differences and how big they are, it can influence some kind of new way of thinking about giving antibiotics constantly, or for exacerbations, big amounts of antibiotics. And also modulator therapy helps by reducing the amount of antibiotics these patients receive. So that could also make a difference, hopefully.
Thank you for your presentation. There were a lot of talk about confounding variables. So my question would be that, statistically speaking, what is a confounding variable and how you try to address it in your research?
Thank you for the question. So confounding are factors that we do not do an analysis on that could potentially influence our results and could cause bias. And one of our biggest confounding, also visible on this first plot, was age. That we accounted for. So different age groups have very different gut microbiota composition. So, for example, infants are way different from 2-year-old or a 3-year-old. But when you reach that age through 3 or 4 years old, it is kind of similar to the adult population by that point. So that is one big confounder that we try to account for. Well, all the other confounding factors that we mentioned before, also the high risk of bias of the articles that we included was a way of a kind of confounding factor.
Thank you.
Thank you.
So just a quick question. There was mention of shotgun sequencing and 16S RNA. And what's the ratio of the studies included, like 50-50 or?
Oh, no. So shotgun sequencing is way more expensive than so the whole genome sequencing is way more expensive than shotgun sequencing. So maybe 2 or 3 studies. 3 studies used whole genome sequencing, so all the others used shotgun method.